Good afternoon to all. I will speak today about modeling of axon elongation using linear element method, modeling by stem cells. And I will tell you that this is a new topic in our center, so this is somewhere beginning of investigation. First, I will tell that axon is part of neuron, and the basic function of axon is to conduct electrical impulses away from the neuron's cell body, and axons make connection with other cell at a junction called synapses, and thanks to special molecular structures, transmit electrical or electrochemical signals across the gap. And this is one picture of neuron, shorter extrude from cell body, represents dendrite, and longer axon. Difference between dendrite and axon is also that axon transmits signal, while dendrite usually receives a signal. And when axon is damaged, then there is no conduction from one part of body to the another, so that axon needs to be repaired, and thanks to stem cells we have opportunity to do that. This is one picture of neurons, and this longer lines represents axon, and I will tell you something about stem cells that are biological cells typical for multicellular organism, and there are two different types. We have embryonic stem cells and stem cells, adult stem cells. Embryonic are isolated in the inner cell mass of blastocyst, and adult can be found in various tissues. Two basic characteristics of stem cells are division, which means that stem cell can divide many times without change type of cell, and most important, differentiation, when from one cell we could get any other cell of organism, including self-renewal. And compatibility of differentiation determines potency of stem cells. And these are named several types of stem cells: titopotent, pluripotent, multipotent, oligopotent, unipotent, in order from highest potency of differentiation to the lowest. So from fertilized egg we get totipotent stem cells. That is a stem cell with highest potency of differentiation, and from them we get blastocysts containing pluripotent stem cells, isolated pluripotent stem cells from inner cell mass, served for cultured pluripotent stem cells. And on the other side you can see hematopoietic stem cell, neural mesenchymal stem cells, which are also formed from pluripotent stem cells, and which gives us all blood cells, neurons, and bones, cartilage, tissues, and other. And on that slide I named several materials which are using in modeling of axon, nerve regeneration, tubular nerve guides, hydrogels, bioactive glasses, and supramolecular hydroglasses. They all have some advantages in modeling, and we choose materials if we have short gaps, then tubular net guides is a good choice, and also if we want to provide synthetically favor nail growth, we will use supramolecular hydrogels. Models, growing axons move through environment via growth cone, which is placed at the top of stem cells, and we accomplish growing by adding some mass, but also mechanical tension could increase axonal elongation. So force generation at the growth cone has impact on at axon elongation, and we model axon as a material with viscoelastic properties. First model was created based on Matthew O'Toole experiment, and they are applied at growth cone force of 200 microdynes. Length is 200 micrometers and radius 1 micrometers, and they change force which applied along axon. But when we model with finite element method, we need to change Young's models through axon to accomplish non-linear elongation of axon. Here is a function for Young's models, which is changed through axon. X represents current position in axon, and L of t is current extension of axon. Eta and G are both constant of viscosity. Eta is constant of friction, adhesion between axon and substrate, and G is axial viscosity. Greater G includes product of axial viscosity and cross-sectional area of axon, which is important in case when we extend axon and if axon tiny. And for that geometric parameters, after 2,800 seconds, we obtained results of elongation about 18 micrometers. This is a simple model, but we need to do that to prove that these equations are properly. And on that picture we have axon exponential elongation. On x-axis we have velocity of elongation, and vertical is initial distance from cell body. We held that distance is 0 if we have interface between soma and neurit, and maximal length of axon is growth cone. So this is 200 elements of finite elements model, and that is initial distance. So if we closer to cell body, we will have smaller deformation, smaller elongation of axon, and the greater elongation is at the growth cone. In case when there is no additional mass to the axon, deformation of axon is almost linear. So we do that in our solver with finite element methods, and compare these results with results from MATLAB, which is obtained Matthew. So we obtained good match for greater time. of simulation. These two curves has a little different values, but they're pretty much good compared. This is after 280 seconds simulation. And also, beside that, adding mass to axon, infinite element method can be modeled by increasing concentration of material. Here is Galvanian equation, partial differential equation of diffusion. There is the diffusion of tubulin. A is active transport of tubulin from cell body. G is constant rate of tubulin degradation. And C is current concentration of tubulin along the length. Beside that, we have some initial characteristics. I said before that when x is equal to 0, we assume that we have some Neuric interface, and x equal L is length of Neuric. That length changes with time, and we have here ordinary differential equation where it can be seen that change of length in time depends on current concentration of tubulin. And we have also two constants, elongation rate and retraction rate. Retraction rate is calculated from level of degradation of tubulin. For model is also important boundary conditions. We have change of flux on the left side and on the right side. On the left side, when x is equal to 0 in contact soma with axon, we have steady flux of tubulin, and on the right side, current extension of length. We will also have change of flux, and this change is proportional to current quantity of tubulin here. And this is just one simple model where we only test this equation. Initial length is shown on top picture, and after 200 seconds we will have some elongation of axon based on change of concentration. So this model is working, but we are currently waiting for experimental results from our partners from Croatia and España. So then we will get this model with real properties, characteristics. For now, equation works. That is all for me.
Thank you very much. Is questions for Milica? Jana?
Just for the stem cells, what did you do? I mean, what do they do with the stem cells? They first differentiate them to neurons and then they measure the...
No. They usually use these stem cells. Just a second.
Meaning the experiment.
Yes, I will tell you. They usually use hematopoietic stem cells or neural stem cell, and they made some kind of scaffold. They called, they have extracellular matrix, and they place axon on that substrate which contains these stem cells, and on certain conditions, these stem cells will have differentiation and the proliferation rate, and they will grow to neural cells or can combine. I understand that it is the procedure.
Probably it's neural or mesenchymal, not hematopoietic, because they make blood cells.
Promise, I don't hear.
No, I'm just saying not hematopoietic.
Yes, yes, because hematopoietic gives blood vessel,
Yeah.
but I find some in literature that they mention also these cells. So I don't know, but I read somewhere.
But this, I'm just asking for this specific experiment. I mean, the measuring of the axon, basically you start using the already differentiated nerve
Yes,
cells.
yes,
So
yes,
that's, okay.
yes.
Okay.
We still don't have any experimental results, but we'll see.
Yeah, just one comment. So we're just in the beginning of this research, and the idea is try to make some kind of simulation for existing imaging. Does it from neural cells or stem cells, doesn't matter. So just some kind of model where we have a lot of picture in time actually, and the idea try to make some kind of evolution this process in time and try to model it. So we need a lot of input still from biologists, from medical doctors regarding this one, and it's also one European project which is still preparing, and we hope we will get it regarding this issue. So I hope we will go in this direction much deeper. So thank you again, Ilse.
Now a recall presentation of Miloš Radović. Miloš is a PhD student at the Faculty of Engineering, where he also works as the teaching associate. He's also a research associate at the Research and Development Center for Bioengineering in Kragujevac. He's engaged in the project of the Ministry of Science and also in the FP7 Art with ICT project. His research focuses on data mining and optimization of mathematical models in bioengineering.
Proveri task.
Okay. My name is Miloš Radović. I work in Research and Development Center for Bioengineering, and I'm going to speak about mining data from the CFD simulations. So first of all, what is data mining? Data mining is a process of discovering some patterns in large data sets, and the major goal, is to discover some previously unknown relationships between the data, and then use these new relationships to make some predictions, to create some prediction model. So the idea of this work is to use some results of the previously done simulations, and then use this data to train our data mining models, and then use the best model to estimate the desired value in a much shorter time. So the point is to save some time, to avoid the expensive simulations. So first problem we were solving is the prediction of the maximum value of the wall shear stress for a model of carotid artery bifurcation. When we speak about data mining, we always must have data sets in the form of inputs and we always must have some inputs and some corresponding outputs. In this case, the output is the maximum value of the wall shear stress, for this model, and the inputs are geometric parameters, which we can see in this picture. And the values, the mean values of these parameters are given in this table. So we have 12 inputs parameters, 12 geometric parameters, and one continuous output value, the value of the maximum wall shear stress. We actually created 1886 different geometries. For all of them, we did CFD simulations. We calculated the maximum value of the wall shear stress, and then we used this data to train the following data mining models. We used K-nearest neighborhood algorithm, linear regression, neural network, multi-line perceptron, random forest, and support vector machine. These are all the data mining models which are able to deal with the regression problem. And this is the way we tested our models. We used leave one out cross validation. We calculated the relative mean squared error, which is given with this formula, and according to the literature, if this error is smaller than 1, then our model can be useful. So here we can see the first results. We have 4 models which have the error smaller than 1, and the best model is the neural network. And since this error is more than 4 times better than, smaller than the next model, we were focused on this model. This neural network is, the structure of this neural network is given here, and it is a multi-line perceptron with 12 inputs, one for each geometric parameter, one output, which corresponds to the maximum wall shear stress value, and this network has one hidden layer with 5 neurons inside of it. And for training this algorithm, we used for training this model we used gradient descent backpropagation algorithm. And after this, we did some model explanation. We wanted to see which of these 12 geometric parameters are the important ones for our model. And on this picture, we have 12 diagrams, one for each geometric parameter, and on the horizontal axis, we have the values of these geometric parameters, and on the vertical axis we have the contribution to the output. And here we can see that the circled one parameters are the important ones. And how did we calculate this? We calculated the contribution by using this simple algorithm. We actually, for every value of every geometric parameter, calculated the contribution. And this is an algorithm proposed by Igor Kononenko and his team from Slovenia. By using this algorithm, we were able to plot these diagrams, and we can see that these 5 parameters are important ones. The other one are more or less flat. So after this, we used only these 5 parameters for training our neural network, and we were even able to reduce the error. We get smaller error. So we can conclude that the other parameters were making some noise in the data set. After this, we continued our work with the prediction of the entire wall shear stress distribution. We wanted now to predict not only the maximum value, we wanted to predict the entire distribution. And now for 2 models, for aneurysm and carotid bifurcation models. We see these 2 finite element models in these 2 pictures, so aneurysm and carotid bifurcation. Now we have 2 data sets, one for carotid bifurcation model, but now we have for the outputs entire distribution of the wall shear stress, not just one value. Now we predict the entire distribution. And for the inputs, we use the geometric parameters, same 12 parameters I was talking a few minutes ago. And now we included the density, dynamic